Yes, all set. Cool. Hi, everyone. I'm Sam. I'm really excited to be talking to you today. I'm going to present our work on topsy-turvy 3D, wherein we leverage pre-computed protein 3D sequence models to predict protein-protein interactions. This is work with Kapil Dakota, Rohit Singh, Lenore Cowan, and Bonnie Berger from MIT and from Tufts. So classical methods like docking take known interactions with proteins with known structures and try to fit them together. This is a really powerful approach, but it's computationally intensive, and crucially, it requires that we actually know beforehand that these proteins interact. The other thing that we could do, given two proteins, is just make a prediction, a binary label of whether or not they interact. And today we're going to focus on a class of sequence-based deep learning methods that take protein sequences and make a prediction of interaction. And we're less concerned about the structure of the complex, although, of course, we want to push more and more in this vertical axis. Yes.
The virtual audience cannot see the screen.
Cool, thank you. Yes, so we of course want to constantly improve our predictions of how proteins interact, but really what we're interested in is really high throughput, scalable prediction of protein-protein interactions. We want to be able to go into a species where there's little or nothing known about the proteins other than their sequences, which, than their sequences, and make predictions of the whole network. And given a network, then we can do network analysis and extract functional modules and pathways. So at 3D-SIG last year, we introduced this method called topsy-turvy. Topsy-turvy combines a method called Descript, which is also a member of this sequence-based deep learning class, uses pre-trained protein language models from Bepler and Berger to make generalizable predictions of protein-protein interaction across species. We combine Descript with a method called Glide. Glide uses a training network and looks at the network topology to predict missing edges. And these missing edges are then used to augment the training for Descript. Thus, we have a method that integrates the top-down network and bottom-up sequence-based approach of protein-protein interactions. But crucially, although we use the full network during training time, topsy-turvy still requires only sequences at inference or at prediction. It's also really important to note that we're not the only people doing this. There's a wealth of methods in this kind of sequence-based rapid protein interaction prediction, including actually rapid was presented also at ISMB last year, I believe. So with topsy-turvy, using the structure information from the Bepler and Berger protein language model, topsy-turvy makes predictions of if proteins interact, but with some information about structure. But we want to take this even further, and that's the question we ask with topsy-turvy 3D. I'm sure I don't need to convince this audience that structure is really important for understanding if and how proteins interact. You can have proteins with very different sequences that will interact in similar ways because the structural conformations they adopt are actually very similar. So in the past couple years, and including this workshop, there's really been this explosion in protein structure modeling, and we want to ask how we can leverage these advances in research to improve sequence-based PPI prediction. And so I want to note that it isn't immediately clear how to actually leverage this existing research. One thing that you could try to do is treat PPI prediction as a purely structural problem and use something like AlphaFold multimer to just predict the structure of the complex of the two sequences. But one potential issue is that AlphaFold multimer is really still intractable, and related methods at this genome scale, all by all of a genome in an unknown species. And we found that there's not, the IPTM scores predicted by AlphaFold multimer might not always be calibrated to the actual probability of interaction prediction. So what about instead of the multimer structure, we just look at single protein structure? There's been this real revolution in single protein structure prediction kicked off by AlphaFold in 2021, but also including RosettaFold, OmegaFold, among a number of other methods. And these methods are really, really powerful, give us really high quality structures. I'm sorry, the slides are jumping all over. I'm not touching the computer. But protein structure prediction is expensive along a whole bunch of axes. It's expensive from a time dimension, right? The amount of time required to train and do inference with these models. It's expensive in an expensive fashion. This is just kind of a back of the envelope estimate for the cost of training AlphaFold and RosettaFold based on their reported compute usage and kind of some Microsoft Azure cloud. These are back of the envelope estimates, but are also almost certainly an underestimate of the total cost required to train these models because it doesn't include hyperparameter tuning and model selection that go into this training. In addition to the time cost and the fiscal cost, there's also an energy cost, right, associated with these models. Again, it's hard to pin down exactly how many pounds of CO2 are used by any particular run, but this benchmark or this paper from 2019 compares the CO2 usage of air travel, of human lifespan, with training of these large transformer models. And these kind of latest generation protein structure models are on a similar order of magnitude of parameters as GPT-2 here. So there's a substantial carbon cost to these structure models as well. The good news is we're a community, right? We don't all have to do the same work over and over again, and we can leverage the structures that exist and these massive community efforts that have gone into not only building these structural models, but computing structures through efforts like the AlphaFold protein structure database, where we now have over 200 million protein structures available to download. So the question we then pose is, okay, how do we take these protein structures that are predicted and available from these whatever fold structure predictions and use them to improve protein interaction prediction with topsy-turvy 3D? But again, crucially, we want to remain scalable. We want to be able to do this at genome scale and predict networks de novo. Okay. So we want to do something really well. We want to do it really fast. We have a good idea where to turn. Again, I'm sure I don't need to convince this audience. FoldSeek, I believe, was presented at this workshop last year. But FoldSeek is this really cool, very powerful tool, actually originally designed for searching for protein structures. The idea being take a structure and encode it in a sequence, and you can leverage the wealth of technology we have on really rapid sequence search, things like MMSeqs, to search for similar structures. The way that they do this briefly is for a given residue, they find a nearest neighbor based on the virtual center, extract bond angles and distances around this residue, and then train a vector quantized autoencoder to learn kind of a latent space of the structure, where each one of these quantized latent vectors can then be mapped to a token in this 3D-I sequence, where I want to note that the tokens of these sequences don't correspond to side chain identity or amino acid identity, but rather the shape of the protein. And I've pulled this figure from their supplements, which I think really nicely illustrates why we think FoldSeek and the 3D-I sequences are useful for our purposes. For a given token here, these are kind of 5 residue backbone pairs at that token in the 3D-I sequence. And although the amino acid identity isn't the same, you can see the structures of the chain here are similar. So the 3D-I sequence is fast. It keeps us in this kind of sequence-based mode, but really imbues a notion of the structure of these proteins. So FoldSeek then is our bridge from the structures that we have available in things like AlphaFold DB and topsy-turvy 3D predicting protein-protein interactions. The approach we take is actually kind of the simplest thing that you could think of, which is given a FoldSeek sequence, or a 3D-I sequence rather, we will one-hot encode it and concatenate that representation to the projection that comes from our pre-trained protein language model. This joint embedding is then passed through the remainder of the steps in topsy-turvy 3D, the outer product, the prediction of an interprotein contact map, and the pooling and summarization that finally leads to a prediction of whether or not the proteins will interact. So like topsy-turvy, TT3D is trained and evaluated in the cross-species setting. We're really interested in making predictions for species where there's very little known about protein interaction in these species. And we do that by leveraging the lot of information that we do know about human protein interactions. So these models are trained on human networks and then evaluated in a number of different species, including mouse, fly, roundworm, E. coli, brewer's yeast. And just a brief technical note, the way that we actually extract the 3D-I sequences for the proteins in our training and test sets is just by querying the AlphaFold DB for exact sequence matches. And actually the FoldSeek folks have already released 3D-I sequences for a lot of the structures in the AlphaFold DB, so we don't even need to compute the FoldSeek sequences, we can just query those directly. For sequences for which there was not an exact match in AlphaFold DB, we've taken a conservative approach and just concatenated an uninformative zero vector. And I'll talk a little bit about in the future work about things we're thinking about in terms of better ways to do this representation, but we took a kind of conservative approach, so we didn't have kind of false positive matches on the 3D-I sequence. And so what we found just with this kind of simple addition of the 3D-I representation to our protein language model representation is that we were able to improve performance on our cross-species setting over topsy-turvy and over Descript. Not only in the AUPR, but what we were really excited to see is that by including this information about structure, we improve our top K precision, or our precision at very low recall. Because we want to have predictions that we're very confident in and that we can go and do biological validation on, we're less concerned about missing interactions that may be there, and we want to err on the side of, for predictions that we do make, being very confident that those are true interactions. And so we were really excited to see that this simple change here allowed us to kind of flatten out the tops of these curves and improve our precision at low recall. So how can we more intelligently leverage structures? The first thing that we thought about is, okay, these kind of autoencoder by fold-seq maps to tokens, but by just using a one-hot encoding of the token, you're actually losing information about dependencies between the states and 3D-I states that might have different tokens but correlate to similar structures. And so one thing we're interested in looking into is actually using the latent vectors learned by the autoencoder as our representations of the 3D-I states rather than a one-hot encoding. And actually now there was this great talk yesterday on Prost T5 actually training a language model that translates from amino acid sequences to fold-seq sequences. And that's, of course, something we'll have to try. But if that works, that's really exciting because now we don't even need to go through structure. We can get these structure representation 3D-I sequences directly from amino acid sequence. In addition to kind of imbuing structure into our inputs and our representations, we want to look into how we can bring structure into our supervision. This is akin to something like template modeling, where given a query pair of sequences, or structures rather, you look into a database for a complex that looks similar, and you say, our query proteins probably interact in a way that looks like this. But because we want to kind of maintain this sequence-based framework, we can use the growing number of solved structures or predicted structures for complexes by efforts like AlphaFold Multimer. And actually, because TT3D makes this intermediate prediction of a protein contact map, supervised directly at the contact map level. So we're not only supervising whether or not the proteins interact, but how they interact. This will hopefully improve both tasks, right? The interaction prediction and the quality of this contact map. So I'd like to thank, of course, my collaborators on this project, Kapil, Rohit, Lenore, and Bonnie. Thank you to the funding agencies, NIH and NSF. I'm happy to take any questions. Also, we have a poster on this work later this afternoon, B44, as well as a poster, B43, on complex, which is our work using protein language models and contrastive learning to predict drug-target interactions. And also GitHub, these are all software packages, open source. Please, we hope they can be useful for you. Please give them a try and let us know how we can help with them. Thanks.
Thank you very much for the talk. Sorry, I have a question about your training regime, because on my slides you said you're kind of training just purely on the single chain models, and on the other side you mentioned the cross-species factors, and you said the learning from the human interactions can be transferred to some other species. So I wonder, did you actually train just on monomeric structure, or you also gathered a data set from human interactions during your training? So I don't know.
So we, all of our data is binary. We're not looking at higher order complexes. And we are not we're not presently incorporating the structure of complexes into our training, just the sequences.
What's the reason that you just use the predictive structure, not the real protein structures like from PDB during your training?
So we're not using any structures right now. I think that's one of the things that we're hoping to look into here. And certainly if a real known structure exists for a pair of proteins, that's preferable to a predicted structure, a really high confidence crystal structure. But we think that if we have high confidence multimer predictions, we could incorporate that into training. But right now we're not using any structures during training. Or any multimer structures.
I didn't quite understand the answer you gave me because I thought you asked about the fact that it's training one organism and tested in another. So, like, that's what you did, right?
Yes, yes, we trained on human interactions and we evaluated on networks from other species.
So how do you know that you're not looking at the trivial case that it's a normal of the same complex?
Oh, like homologous sequences? We do some filtering on sequence similarity before we split the data set. We use CD hit and filter to 40% sequence similarity.
Any more questions? We still have some time. Yeah.
Could you say a little more about how you're training with purely sequences and incorporating fold-seq? I think that some of the questions imply that people are missing that point.
Yeah, so the actual input to the TT3D model and to topsy-turvy before it is a sequence. Because now we have these we have these predicted structures for sequences, we can find the structures for the sequences, use fold-seq to find a corresponding, or rather use the AlphaFold database to find the structure and turn that into the 3D-I sequence. Is that what you're asking?
And then how are you combining?
Oh, yeah. So the amino acid sequence is encoded with the pre-trained protein language model, and we do kind of a projection on that to reduce the dimension. And we just concatenate that representation with a one-hot encoding of the 3D-I sequence. And we have this kind of joint embedding that has both information from the protein language model and from the 3D-I sequence.
And what do you get from the 3D-I sequence again?
The 3D-I sequence helps us understand, it's a kind of a compact representation of the protein structure rather than trying to encode the individual atomic positions or like a voxel representation. We just have this really compact, easy to work with, but very information-rich sequence representation.
Any more questions? I find it quite interesting that all these methods that are well known now in the field were all presented at 3D-SIG in the years past, and how you cross-pollinated with things that were shown this year already, and maybe we can see next year how it impacts.
Yeah, we're definitely thinking about future work here, and there's been a lot of really interesting ideas that we've seen so far and probably are yet to see.
Very good. Well, thank you very much.